Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last class we started our discussion on the first next generation sequencing technology, which was Roche 454. So, in this class we will continue our discussion on other next generation sequencing technologies that are available today, and especially we will be focusing on the Illumina sequencing by synthesis method. Okay, so, this is the agenda for this class. So, we will talk about this Illumina sequencing by synthesis technique and how this works. We will talk about single end and paired end sequencing. So, let us start. So, these are the keywords that will come across. One is reversible termination, bridge amplification and short and long read sequencing. Okay. So, in Illumina sequencing is done on a flow cell, not picotetraplate, but it is very similar idea. So, what you have there is something like a plate, right, and you can find lanes here. So, you will see this, if you see a picture of this, right, you will have these lanes, and where you can have these DNA molecules that will be bound in these lanes. Okay. So, we will talk about this in a bit more detail. So, you have a plate like structure and then you have lanes inside which all this sequencing reaction will happen. Okay. So, this is called a flow cell. Okay. Now, coming to the library preparation. So, this is the typical library preparation as we have discussed already. So, we have DNA fragmentation starting from the genomic sequence. You have adapter ligation, adapter A and B that have been ligated to these fragments, and you have amplification and sequencing. Now, we will talk about this amplification step. We have seen in Roche 454, this amplification step actually requires something called emulsion PCR, right. So, this is specific to 454 sequencing. In this method, in Illumina, this method is called bridge amplification, and we will see in a moment why this is called bridge amplification. Okay. So, let us look at this, right. So, as you have seen, so what happens in this flow cell, inside this flow cell, you can imagine, right, is you again have some DNA molecules that are complementary to these adapter sequences. Okay. So, like 454, you have certain sequences, DNA probes that are complementary to adapters. So, what happens now is that this DNA molecule with adapters will come and hybridize, right. So, here is again let us say this will come and hybridize. Now, this means you can generate or amplify these fragments, okay, using DNA polymerase. Okay. You can then amplify these fragments using DNA polymerase. Yeah. So, what will happen is that once this is amplified, these molecules can then be released, right. So, once these fragments are extended starting from this complementary sequence, you can release these fragments, these DNA fragments. Okay. So, what you have in after the end of this process is something like this. Okay. So, we have these DNA fragments, right, that are generated on this plate. Okay. And again we can imagine you have millions of them on this plate. Now, what happens is that again you need to calibrate this, the number of DNA molecules that you will allow to hybridize on this plate, is that in the next step there is this bridge amplification. So, what is this idea? What is the idea behind this bridge amplification? So, you have seen this emulsion PCR. This is a clonal amplification, right. So, where a single molecule actually generates multiple copies inside these emulsions. Okay. Why do you generate these many copies? Because for the signal detection that is happening, we need a lot more sequencing process. So, these detectors in 454 or in Illumina, they cannot directly identify the signal from a single molecule. Okay. So, if you have clonal molecules, they will be synthesized, right, the complementary strands to those molecules will be synthesized at the same time, because they contain the same sequence, right, and that signal comes as the signal from that well in 454. Similarly, in Illumina you need to generate these clonal molecules, right, which we call as clusters. Okay. So, how do you generate these clusters? We do this by a process called bridge amplification. Now, what happens? We have these amplifications happening, right, here, right. We have generated these DNA fragments on this flow cell, but surrounding that you will also have some DNA probes that will be complementary to the other part of this DNA fragment. So, remember the other part contains this other adapter, right. So, if you imagine this is adapter A, this is adapter B. So, here adapter A, adapter B, right. So, the other part contains adapter B. Again we know the sequence of this adapter B. So, you can have these DNA probes or oligos on the flow cell that are complementary to this adapter B. Okay. So, if that is the case, then what will happen in this scenario is that we will focus on one molecule just here. So, this will come and it will fold here and it will kind of show something like a bridge structure. Okay. So, this will actually come and bend and will form something like a bridge structure. So, you can see this kind of structure, right. So, this is the bridge structure. So, once you have this bridge structure here, you can again synthesize this, right. You can amplify this using DNA polymerase and DNTPs, right, normal amplification, and this is called the bridge amplification, right. Because you have this bridge like structure, this is called the bridge amplification. Okay. So, the idea again is to generate multiple copies of the same clone, right. So, because you are generating this bridge, right. So, you can imagine now this happens, this is amplified. Now, this molecule will bend, and if there is another complementary adapter here, it will bend and form this bridge, and there will be amplification again, and this process is repeated, right, across all the adapter complementary sequence that are present in the flow cell, right. So, this is a very small region where this is happening, and you can imagine this is happening in parallel for millions of positions in the flow cell. Okay. So, each position will contain clonal copies of the same molecule, okay, and this forms the cluster. Okay. And this cluster is where the sequencing reaction will take place and the signals will come, right, and the signal that will come will be the average signal, right, and this will be much stronger because the signal is coming from multiple molecules at a time, and these molecules contain the same sequence. So, they are expected to give out same signal during the sequencing process. Okay. So, we have now generated these clusters and we can now proceed to the sequencing step. Okay. So, here the sequencing step again is a sequencing by synthesis method or SPS, which means we need to synthesize the complementary strand using DNA polymerase and DNTPs. Now, again the DNTPs that are used in this process are not normal DNTPs, these are modified DNTPs, and in such a way that it allows for reversible termination. Okay. So, we have seen dideoxy termination in case of Sanger sequencing. Here we have reversible termination, which means once these bases bind, right, these modified bases bind to the complementary strand that is being synthesized, they will block the synthesis process, but with the subsequent wash and release step they can be removed again. Okay. So, it is not a irreversible termination, right. These terminators, their modifications are such that these termination sequences or termination groups can be removed from the DNTPs and the synthesis can go on. Okay. So, with this in mind, right, so we also have something called fluorescently labeled DNTPs there. So, you have special DNTPs that contain these terminators as well as fluorescent labels. Again, each DNTP is labeled with unique color, right. So, unique fluorescence, and all these bases are present in every cycle of sequencing reaction. Okay. So, since each DNTP has its own unique color, they can be identified very easily by the detector during the synthesis process. Okay, so there are multiple steps. The first step is the base addition, I will illustrate in a moment. So, where these DNTPs are added, right, and then the second step is the signal detection, where the detector, there is an optical detector that can read the signal, right, the fluorescent signal that is present and identify the base, right. And then the third step is cleavage of reversible terminator and fluorescent dye. So, the dye from the already incorporated base is washed out. Okay. So, let us draw this, right. So, you can imagine now, let us say you have this on the flow cell, we just focus on one single molecule, right. We have the adapter here, we have primer, right. Here the primer is complementary to the adapter, let us say, and we do the synthesis in this direction, right. So, what we have is polymerase, we have DNA polymerase, and we have modified DNTPs. Okay. So, as I have just mentioned, these are reversible terminators, right. So, what happens, let us say there is addition of A, okay, and it has a fluorescent signal F, okay, and this may be a unique one, right. So, again if you can imagine, right, this probably, let us say it is red, this fluorescence. So, once this base A is added, right, so the synthesis process will stop, right. We are using this modified DNTPs only, right, no normal DNTP. So, the synthesis process will stop, but there is a fluorescent signal which will be detected by the detector, okay, and this will identify, okay, there is an addition of A. Okay. So, the sequence then reads as A. Okay. In the next step, this terminator and the fluorescence is washed out. Okay. So, these groups are cleaved, so that they are washed out and there is no fluorescence. So, this is gone now, the terminator is also gone. Then let us say the next base is T that is added. Okay. And again there will be some fluorescent signal, again something different compared to A, but it will also stop the synthesis process, and there will be a detection of signal. So, T, then you have the washing step, right, this F is gone, right, there is cleavage, and also the terminator is gone, and the next base will come in now. Okay. So, this process will go on, right. So, you will have now G, C, etc. Again these all the bases are present. So, depending on the complementary sequence, these bases will be incorporated by the DNA polymerase. Now, one thing you probably have noticed now, right, that this base addition is happening one base at a time. Okay. There is no multi base addition because of this terminator. Okay. So, even if you have, let us say, multiple A's present, they will not be incorporated at once. Okay. They will be again incorporated one by one. Okay. So, what is the advantage then is that this will eliminate any problem with homopolymeric stretch, right. So, even if you have, let us say, very long homopolymeric stretch, 25 base pair, 30 base pair, or even longer, you will have no issue in determining the exact length, because you are detecting signal for one base at a time. Okay. And you can now imagine, right, this process is happening across millions of clusters. Okay. So, we have just seen for one single cluster here the process, but this is happening across millions of clusters in the flow cell, and in parallel you are sequencing millions of fragments, you are detecting these signals one base at a time. Okay. So, that is why this method is very accurate, right. So, you are getting this done in one base at a time process, and this makes this process very accurate. Okay. So, here are some key points that you need to remember. So, we have just mentioned that all DNTPs are present in each cycle, right. So, all are there, and whichever is found to be complementary is incorporated, and this is not an issue because the detector can detect each of these bases by their tags, right. So, because each base has its own tag, so the detector, the optical detector is there, it can actually identify these individual bases very easily. And one point that we have seen is that one base is added in one cycle. Okay. You do not have multi-base additions at once, and this is in contrast to 454 sequencing, where there is no limit, right. So, you can have 20 base addition, right. So, if you have 20 S, they will be added one after another very quickly, and then the signal will be read out, right. So, which will be the total signal intensity or the light intensity that we get. Here we are getting signal for one base at a time, and this makes for a very accurate sequencing process. What it also means, the number of cycles will determine the read length, right. So, you are controlling the number of cycles of this base addition, signal detection and cleavage, right. So, this is we have talked about these 3 steps, right. So, we have step 1, 2, 3, right. Here is the addition, base addition, we have detection, and then you have the cleavage. Okay. So, you have these 3 steps in one cycle. So, this constitute a cycle, right. So, you have these 3 steps, addition, detection, cleavage. You can decide how many cycles you want, right, by controlling the cleavage step, for example, right. If you do not want any more cycles, you just stop there, and there will not be any more synthesis after that. All right. So, you can, the number of cycles that you allow will determine the read length that you get. Okay. So, in Illumina it is typically 150, 200 or 250 at most, right. So, you can control between 50 to 150 to 250, right. So, you can decide, okay, we will stop this after, let us say 150 cycles. Okay. So, depending on the machine that you are using. Again, some machines can handle 250 cycles, some machines will give only 150 cycles. The other outcome is that all reads are of same length, right. So, you can imagine this process happening across these clusters, all these clusters, millions of them, and everywhere there is this reading of one base at a time, right, and you are supplying this or doing this cycle thing for each of these clusters, right, similar manner. So, all reads that are generated in Illumina will be of same length because of these reversible terminators. Okay. So, we understand how these actually impact the sequencing process. So, in comparison, Roche 454 you will get reads of varying lengths, right, again because in some cycles you might have, let us say 10 A's added, and the next one you have 5 G's added, and this number is different from different fragments, right. So, you get reads of different lengths, but in Illumina you get reads of the same length. Okay. So, here is an overview of the whole process that you can see. There is a very nice animation of the whole process that you can see, starting from the library library process, the cluster generation, bridge amplification, then the sequencing, okay, and you will get a very nice idea about the whole process. So, one thing I also like to mention is that these sequencing methods are also changing very rapidly. Okay. So, here is one example of the sequencing chemistry in Illumina that is also evolving, starting from 4-channel chemistry, where you needed fluorescent signals to detect these 4 bases. It has now come down to 1-channel chemistry. So, it needs only one signal to detect even 4 bases, right. So, this actually makes for even faster sequencing, because you do not, in 4-channel chemistry you need to take images, right, 4 images in 4 different wavelengths to identify these 4 different fluorescents, right. So, for each cycle you have this image acquisition, and that actually slows down the whole process. In 1-channel chemistry you can take these images in just in single channel, right. So, that is a much faster process for the sequencing. So, as you can see, right, as the number of cycles increase, this actually improves in time part, right. So, we can make these processes, the sequencing process, much faster. Now, one of the key ideas in Illumina is called paired end sequencing. So, we will talk about that now. So, there is something called single end sequencing and something called paired end sequencing. Okay. So, in single end sequencing you have sequencing from one end of a DNA fragment, right. So, you can imagine this is a DNA fragment in yellow, and we have these adapters, adapter 1 and adapter 2, and we can decide that we will just sequence from adapter 1. Okay. So, we add primers in such a way that only we sequence from one direction. All right. So, this is will be your single end sequencing, right. So, we are sequencing from just one end of the DNA fragment that we generated. But in Illumina as well as in 454, you can actually sequence from both ends, right. So, it just requires these primers that are complementary to these adapters, right. So, this is of course not happening at once, right. So, what happens is that you should remember this synthesis process in Illumina, right. So, you have synthesis starting from one end, and then this will continue towards the other end, right, or if you stop the number of cycles, that is it, it will stop the sequencing action, and then you can release the strand that has synthesized, and you can start sequencing from the other strand, right. So, from the other side. Okay. So, this is how the paired end will happen, right, paired end sequencing will happen, that you will sequence from both ends of the same DNA fragment, right, one after the another. Okay. And this actually is very useful in many cases. As we will go along, we will see why we actually prefer sometimes to do paired end sequencing. Okay. So, one of the first advantages of paired end sequencing is to actually it allows detection of genomic rearrangements. So, what do you mean by genomic rearrangements? So, you can have something like this, right. So, let us say you have these two DNA fragments or chromosomes, and maybe there is some sort of exchange between these, okay, between these two chromosomes. Okay. And this will give rise to, let us say we mark them in different ways. So, this will give rise to something like this, right. Okay. So, once you have this kind of chimeric chromosomes, so this will be something like this. Now, if you let us say you are sequencing and trying to identify the genomic changes that has happened in the sample, right, and if you employ let us say single end sequencing and you sequence this part here. Okay. Now, if you are sequencing this part, you have no idea that this genomic rearrangement has happened, okay, with single end sequencing. But if you are using paired end sequencing and you have one part here and the other part here, then you know, right. So, one part is coming from this fragment or this chromosome, and this part is coming from this fragment or this chromosome, right. So, this paired end sequencing will help us in detection of genomic rearrangements. We will see this in much more detail when we actually analyze these variants, etc., and how we actually detect these variations. Okay. We can also have better mapping of repetitive sequences in a genome. So, you can imagine, right, you have this genome sequence and you have often these repeat sequences present. Okay. This is quite common in human genome as well as in other genomes. So, you have these repeat sequences, means they are identical in sequence. Okay. Now, let us say you get some read data, okay, from let us say matching this region, but you cannot be sure whether this read comes from this region or this region, right. They are identical in sequence, and you have got this read, but you do not know whether this read was generated from this region of the genome or that region of the genome, right. So, this is something that is not possible if you are just doing single end sequencing. Now, imagine you are doing paired end sequencing. So, you have one here, right, and the other part you are also sequencing, and that falls here. Okay. Now, this pair, right, this is unique, right. So, this region is unique, okay. There is no repeat sequence here. Since this region maps to this position, this region there is a match with this location of the genome, we can now say, okay, this read comes from this repeat only, right, not from this one. Okay. So, having this pair actually allows us to resolve these reads coming from repetitive sequences. Okay. So, again we will illustrate this in much more detail when you go into the analysis part. And then we can also identify gene fusions, right, as we have just seen, that for genomic rearrangements the idea is very similar. So, you have gene fusions, two different genes fused together in one place, again with sequencing only one part of this gene fusion, you have no idea that this gene fusion has happened. But if you have paired end sequencing, you will see one part coming from one gene and the other part coming from another gene, right, and this can give you an idea about this gene fusions. Okay. So, and then finally, we can also detect mutations more accurately. So, this is actually quite simple. So, imagine this fragment, we are sequencing from both ends and we generate these reads. Okay. And if there is any overlap between these two parts, right. So, we are sequencing from both directions, and we do that sometimes also in Sanger sequencing, right. We sequence a DNA fragment from both directions, and in this overlapping part, right, let us say we find there is a mutation here, there is a change, right, from let us say some base to another. But the other read or the pair here does not show that change. So, it will then tell us, okay, this is probably a sequencing error, right. So, although sequencing error what you have seen is actually low, this happens, right. So, here we can then identify, okay, this is a sequencing error that has happened. Okay. So, in this way we can actually double check the data, right, that we are getting, and this helps us in getting more accurate picture of the mutations, and this is something we do in also in Sanger sequencing. So, there are several sequencers, again varying because of their throughput and the kind of capability that they have, right. So, these are some of the names. You can have minisec, misec, etc., nexsec sequencers. These are the principle of sequencing is same. We follow the sequencing by synthesis method, only thing is that their throughput is different. Okay. So, here is some idea about the throughput that you get from this machine. As you can see, they vary a lot, starting from 1.2 gigabases to 330 gigabases, going for nexsec 1000 to 1000. Again, depending on your requirement, you will choose the right sequencer. The maximum read number is also given. As you can see, you can get 25 million reads. Again, that is a huge number, right, up to 1.1 billion in nexsec platform. Okay. So, these are really big numbers, and which means you can actually work with really big genomes. The maximum read length, right, so that you can get from these sequencers is that 2 into 150. So, 2 means it is paired end. So, you can get paired end into 150 means at most 300 bases that you get from the sequencers. Okay, again there is sequencer specific limitations, there will be problem with the signal detection, and there will be more sequencing errors if you go beyond this. So, these sequencers, most of their applications are in small whole genome sequencing, targeted gene expression analysis, where we want to look at gene expression of only a specific number of genes, microRNA, small RNA analysis, and 16S metagenome sequencing. And then also have production scale sequencers. These are called nexsec 6000 and novasec X, and these are the outputs that you get, and you can see these are even more number of bases that you are getting out of the sequencers, going up to 16 terabases. And maximum read number also increases proportionally. Now you are in the billion levels, right. So, 20 billion, 26 billion, and the read length is again more or less same, 2 into 150, but in case of novasec 6000 you can go up to 2 into 250, again paired end 250 base each. So, up to 500 base pairs. And these production scale sequencers, they have applications in large genome sequencing, right. So, if you are working in big genomes, you will use these sequencers. For transcriptome analysis, where you want to look at gene expression of all genes in the genome, not just targeted analysis, and these can be also used for methylation and chromatin analysis and single cell profiling. The advantages of Illumina sequencing platform is cost effective as compared to Roche 454, and this has actually been the reason for success for Illumina. One of the reasons is very accurate, is even higher than 454, because this has addressed the error in the homopolymeric stretch as you have discussed. So, it is about 99.9% accuracy that you get, and really high throughput. We have seen the numbers, and this is suitable for large scale sequencing, and it is very cost effective, and that is what made it the most widely used technology today, because of all these points together. There are of course some drawbacks, right. So, this is still quite expensive compared to newer NGS techniques, which we will discuss later. This is because of the use of the enzymes, etc. This becomes very expensive. The capital cost is quite high because you have these optical detectors and they have to be very sensitive. So, the cost of the instrument is actually already quite high, and also the running cost because you are using enzymes and modified nucleotides for the sequencing process. It is not cost effective for small scale experiments because you can see like the numbers are really large, the number of reads that you are getting. So, if you have only 1-2 samples, you do not want too much data, then this is not the method that you will use for your application. And one of the major drawbacks is that we get only short reads which is up to 2 into 250 base pairs. So, to address this, Illumina has developed something called long read sequencing technology. So, this addresses the short read issue and get reads of length 5 to 7 kb, and this is generated by something called landmarking. Okay, so, these long DNA fragments are actually first landmarked and then sequenced. So, this is the schematic of something like this, right. So, you have a long DNA fragment, you have landmarked them which are shown in different colors here, these regions using some sort of tagmentation or tagging, right, again using something like a transposes also. And then you can amplify these fragments, you can sequence these fragments separately, and then you can assemble them. Because of these tags that you have, you can identify okay, which fragment will, aligns with which fragment, right, which fragment comes after which fragment. So, you can then assemble them into long reads. So, these are the references that we have utilized for this class. So, to summarize, we have seen that Illumina sequencing utilizes sequencing by synthesis method and based on reversible termination technology. So, this actually allows for very accurate sequencing, and here the base calling and detection is based on optical detection of the fluorescent signal that is out there, and again this chemistry is evolving with time. We get very highly accurate data, but the reads are actually short. And of course, to address that, Illumina has developed something called long read sequencing, and that overcomes the limitation of short reads. But again, this requires multi-step preparation, which is quite complex. You have seen you need this landmarking process, etc. That is it for this class. Thank you.